This video summarizes research investigating the function of mRub 3013 and whether it has a paralogous relationship to mRub 1477 and/or mRub 0224. The project title is An Investigation into the Relationship between mRub 3013, mRub 1477, and mRub 0224. Are they paralogs? The organism of interest for this project is Meiothermus ruber, which is a gram-negative rod-shaped thermophilic bacteria. On the right of the screen is an electron scanning microscope picture of M. ruber. It can grow in temperatures from 35 to 70 degrees Celsius and has an optimal growth temperature of 60 degrees Celsius. M. ruber produces a red pigment and is found primarily in natural hot springs and artificial thermal environments. It was selected for this project because it is part of the Genomic Encyclopedia of Bacteria and Archaea, or GEBA project, and previous research has shown that it has genes orthologous to those in Escherichia coli K12 MG1655, which is the model organism for this research. In approximately 50% of bacteria and nearly all archaea, there is a bacterial defense mechanism called a CRISPR-Cas system. The system is similar to adaptive immunity in mammals and allows for the bacterium to build up a memory against previous viral infections. CRISPR stands for Clustered Regularly Interspaced Palindromic Repeats, and Cas stands for CRISPR-associated proteins. The CRISPR array is an operon composed of the Cas genes and a leader sequence, followed by palindromic repeats separated by spacers, which are derived from DNA of previous infections. There are 3 steps in the CRISPR-Cas system response: spacer acquisition, also known as adaptation, cRNA biogenesis, and interference. Figure 2, Panel A, shows a visual representation of these steps. Spacer acquisition involves cutting up foreign DNA from an invader and adding it to the CRISPR array. New spacers are added directly after the leader sequence, and a new repeat sequence is made for each spacer. cRNA biogenesis is the transcription and maturation of CRISPR RNA. Each mature cRNA contains a whole spacer and part or all of a repeat sequence before and after the spacer. Finally, interference occurs when the CRISPR effector complex, composed of Cas proteins, binds to the mature cRNA, which guides it to complementary foreign DNA. This ensures the effector complex targets and degrades foreign DNA and not self-DNA. This mechanism effectively shuts down an infection by a viral attacker. There are 6 types of CRISPR-Cas systems. The best studied are types 1 through 3, and types 4 through 6 have just recently been discovered. Type 1 is further broken down into subtypes A through F, and of these, type 1E is the best studied. Figure 2, Panel B, shows the Cas genes that are characteristic of each type of CRISPR-Cas system. Cas1 and Cas2 are universal between all types, and each type has a particular hallmark Cas gene. For example, the hallmark gene for type 1E systems is Cas3. This research project focused on Cas1, which is one of the 2 universal CRISPR-Cas genes. The figure at the bottom of the screen shows the location of Cas1 in the E. coli type 1E CRISPR operon. Studies have shown that Cas1 is essential for spacer acquisition and must complex with Cas2 in order to successfully add new spacers to the CRISPR array. Studies have also shown that Cas2 is not as essential as Cas1 in the addition of spacers. Mutations in the Cas2 active site have little to no effect on the rate of spacer acquisition. In contrast, any mutation to the active site of Cas1 shuts down spacer acquisition almost completely. In the Cas1-Cas2 complex, there are 4 Cas1 proteins and 2 Cas2 proteins. 1 Cas1 dimer is on each side of the Cas2 dimer, and the Cas1 dimers do not interact with each other. How exactly the Cas1-Cas2 complex adds spacers to the CRISPR array is not completely understood and still requires more research. A paralog is a gene that arose through a gene duplication event, resulting in 2 of the same gene. Paralogs can also result from horizontal gene transfer. However, this accounts for a very small proportion of known paralogs. Paralogs are important sources of genome evolution and can evolve in different ways. First, it is entirely possible that both genes will retain the same function. This is unlikely as mutations accumulate, but possible. Another possibility is that the duplicated gene will evolve a novel function to fill a new role. If the original gene carried out multiple functions, it is also possible that the new paralogs will evolve to specialize in one of those functions and divide the original gene's roles. Finally, the duplicated gene may evolve to be non-functional or disappear altogether. Observing paralogs that remain over time provides insight into which genes are most important. 3 functional groups with high rates of paralog conservation are metabolism, transcription, and defense mechanisms. CRISPR-Cas systems are considered prokaryotic defense mechanisms and may have higher rates of paralog conservation. In the M-Ruber genome, we see 3 different genes for the Cas1 protein. In the lower right-hand corner is a picture of the KEGG output for the M-Ruber CRISPR-Cas system, which confirms that mRub3013, mRub1477, and mRub0224 are all Cas1 genes. So, are they paralogs? First, we have an NCBI protein blast alignment between mRub3013 and E. coli B2755, which is widely documented to be Cas1. The query is mRub3013 and the subject is B2755. This figure shows that there is a 40% identity score, indicating that 114 of the 284 aligned amino acids were a match or chemically similar. This alignment has an e-value of 2 times e to the negative 75th, so it is highly significant. These are the NCBI protein blast alignments between mRub1477 and B2755, and mRub0224 and B2755. The mRub1477 alignment has an identity score of 29% and an e-value of 3 times e to the negative 7th, so it is significant. The mRub0224 alignment has an identity score of 34% and an e-value of 5 times 10 to the e to the negative 10th, so it is also significant. These graphs each show any predicted membrane-embedded alpha helices in the structure of each protein. Red peaks indicate a predicted membrane-embedded alpha helical structure. For B2755, mRub1477, and mRub0224, there are no predicted alpha helices, meaning that their proteins are likely localized to the cytoplasm. The graph for mRub3013 shows 2 predicted alpha helices. However, they have low probability and the protein is most likely still localized to the cytoplasm. This table summarizes the data pulled from multiple databases. The first row shows the NCBI protein blast alignments results from earlier in the video. The second row shows the hits each gene pulled from the conserved domains database. All 4 genes pulled COG1518, which is the functional group containing Cas1 proteins. The third row is each gene's hits from the TigerFam database, and these are a little different between genes. Both 2755 and mRub3013 pulled Tiger 03638, which is the TigerFam grouping for Cas1 proteins from or closely related to those found in E. coli. mRub1477 pulled Tiger 03640, which is the Cas1 DVULG category. This grouping contains Cas1 genes from type 1C CRISPR-Cas systems. mRub0224 pulled Tiger 00287, which is another Cas1 family. Row 4 contains the PFAM hits for each gene. All genes pulled PFAM001867, which is the PFAM family for CRISPR-associated Cas proteins. PDB pulls proteins with similar structures from its databases, and these results are in the 5th row of the table. B2755 pulled 5VVK, which is the structure of the Cas1-Cas2 complex when bound to a mimic ligand. mRub3013 pulled 3NKD, which is the CRISPR-associated Cas1 protein found in this project's model organism, E. coli K12. mRub1477 pulled 4WJ0, which is the CRISPR-associated endonuclease Cas1. mRub0224 also pulled a CRISPR-associated endonuclease Cas1 structure, but its tag is 4N06. The final row of the table contains the p-sort B predictions for the localization of each protein. B2755 and mRub3013 are both predicted to be localized to the cytoplasm, while the predictions for mRub1477 and mRub0224 were unclear. These are the chromosome maps for each gene with B2755 on top, then MREB3013, MREB1477, and MREB0224 on bottom. Each gene is indicated with a red arrow and all appear to be part of an operon. They are all followed by a Cas2 gene. B2755 and MREB3013 are both preceded by a CSE3 gene. MREB1477 is preceded by a Cas4 gene, and MREB0224 is preceded by a gene that is simply labeled as a CRISPR-associated protein. Though they all appear to be part of operons, they are operons of varying structure. This figure shows a comparison of the gene neighborhood for MREB3013 and its orthologous genes in evolutionary similar species, with each Cas1 gene indicated in red. There is a high amount of conservation of the gene neighborhood surrounding the Cas1 genes and a large amount of rearrangement surrounding them, suggesting a conserved operon structure. This figure is the same as the previous one, but is built with MREB1477 and its orthologous genes in evolutionarily similar species, with each Cas1 gene indicated in red. There is a high amount of conservation of the gene neighborhood surrounding the Cas1 genes, and there is a large amount of rearrangement surrounding those, suggesting a conserved operon structure for MREB1477 as well. Finally, this figure is a gene neighborhood comparison for MREB0224 and its orthologous genes in evolutionarily similar species. Each Cas1 gene is again indicated in red. There is not much conservation surrounding the Cas1 genes as seen by the differences in each gene neighborhood. This indicates that if MREB0224 isn't an operon, it is not one that has been highly conserved over time. This last figure shows phylogenetic trees built from species with highly similar Cas1 genes to the input gene. In every tree, MREB1477 and MREB0224 are more closely related to each other than they are to MREB3013. This indicates that if MREB3013 does have a paralogous relationship with MREB1477 and MREB0224, the gene duplication event happened a long time ago, and there has been a lot of divergence since then. A more likely explanation is that MREB1477 and MREB0224 are paralogs of each other, but are not directly related to MREB3013 in any way other than their function as Cas1 proteins. To summarize, this research project confirmed the orthologous nature of MREB3013 and B2755. They are both Cas1 proteins in the type 1e CRISPR-Cas systems. Though it is clear that each mRuber gene codes for a Cas1 protein, the relationship between the three genes is still unclear. The data suggests that MREB1477 may be part of a type 1c CRISPR-Cas system, and it is unclear whether MREB0224 is in an operon at all. What is obvious, however, is that MREB1477 and MREB0224 are more closely related to each other than either is to MREB3013. Future research should investigate levels of each mRuber Cas1 protein to reveal which are expressed more frequently, and should further investigate each gene and whether they would function if engineered into a model organism such as E. coli. Finally, future research should work to expand the understanding of the relationship between each mRuber Cas1 gene, as their relationships are still unclear at the conclusion of this project.